Hello and welcome back to the Biochemistry for Health Sciences channel. In today's video, we shall continue our discussion on the starvation state. In video 5.11, we gave an overview of the three metabolic states that humans go through. In videos 5.12 to 5.21, we talked about the post-prandial state, during which we discussed various synthesis pathways. Then we discussed early starvation state in videos 5.22 to 5.25. Glycogenolysis was discussed in video 5.22, and fatty acid beta-oxidation was discussed in videos 5.23 to 5.25. In the last video, video 5.26, we discussed the late starvation state, more specifically the pathway called gluconeogenesis. In this video, we are going to try to answer this question: what happens as we go from having our dinner to not eating for many days? Okay, so what happens as we go from our dinner to not eating for several days? Before we answer this question, we should remember two very important points that we emphasized in our previous videos. So one is that all cells, all the cell types that we have, the 200 or so cell types, must continuously make ATP. We do not store ATP, so that means the cells need to continuously make ATP. The second important point to remember is that while most of your cell types can use either glucose, if it's available, if glucose is not available, they could use fatty acids to make their ATP. However, the brain and the red blood cells do not have a choice. They can only use glucose. So this is a very, very important point to keep at the back of our minds as we go through this understanding of the starvation process. So since the brain and the red blood cells can use only glucose, we have to ask ourselves a very important question as we go through the various metabolic states. And that is, what maintains our blood glucose concentration greater than 40 to 50 milligrams per deciliter? This is the minimum amount we need so that our brain and our red blood cells can extract sufficient glucose from the bloodstream to make their energy for their survival. In the post-prandial state, as we discussed in a previous video, what maintains our blood glucose greater than 40 to 50 milligrams per deciliter is the glucose that we absorb from our diet. This glucose is more than enough not only to feed our brain cells and our red blood cells, but there is sufficient glucose to feed all the other cells. So when we have a carbohydrate meal, there is really no problem in maintaining our blood glucose to feed all of these cells. But what about the starvation state? For example, early starvation when we are not eating. In that case, as we saw in the previous video, it is our liver glycogen. The process of breaking down glycogen to glucose, what we call glycogenolysis, will maintain our blood glucose so that we can feed the brain cells. Similarly, liver glycogenolysis will maintain our blood glucose so that we can feed our red blood cells. And while the brain and the red blood cells are using up glucose, the rest of the cells will be using fatty acids for their ATP. As we shall see in today's video, if we do not eat for more than about 24 hours, all our glycogen will be gone. So there's no more liver glycogen. So now, what is going to maintain our blood glucose at more than 40-50 milligrams per deciliter? And the answer is this synthesis of new glucose that we talked about in the last video, gluconeogenesis. This will be carried out by the liver and the intestine initially, and then later in starvation by the liver as well as the kidneys. And as we shall see, while gluconeogenesis from the liver and the intestine is going to feed the brain as well as the red blood cells, once again, like in early starvation, we will have fatty acids, we'll be feeding the other cells. However, as starvation continues, we see a very interesting thing happen, and that's the formation of another fuel molecule called ketone bodies. And we shall see that these ketone bodies now can also feed the brain besides the glucose that is coming from gluconeogenesis. However, for the red blood cell, only glucose is the fuel that it can use for ATP, while for the other cells, they can use fatty acids or they could use ketone bodies. So with this background, let's now look at the starvation state. We're going to start with early starvation that we talked about in the last video, and then we're going to gradually proceed to the later stages of starvation. So during early starvation, the major metabolic pathways that happen would be glycogenolysis, breakdown of liver glycogen, gluconeogenesis, synthesis of new glucose, and lipolysis, breakdown of triglycerides into fatty acids and glycerol. That happens in adipocytes, and then eventually the fatty acids can be used for energy by beta-oxidation by all cell types except the brain and the red blood cells. So after a meal, your blood glucose goes up, and as your blood glucose goes up, your blood insulin levels go up. And as the insulin levels go up, they stimulate the absorption of glucose into cells, and your blood glucose goes down. And as your blood glucose goes down after a meal, your insulin levels will also go down. So a few hours after a meal, we will see insulin levels are going down, while your glucagon levels, glucagon is a hormone secreted by the alpha cells of the pancreas, while insulin is a hormone secreted by the beta cells of the pancreas. So we'll see insulin levels going down while your glucagon levels go up. So this will give us a low insulin-glucagon ratio. Your insulin is going down, your glucagon is going up, so the ratio will be low. Now this low ratio of insulin-glucagon is going to stimulate glycogen breakdown in the liver, gluconeogenesis in the liver, as well as triglyceride breakdown in the adipocytes. So in early starvation, your blood glucose level will be maintained mostly by glycogenolysis and a little bit by gluconeogenesis, and there will be a lot of lipolysis in the adipocytes So your concentration of fatty acids in the bloodstream increases so that the fatty acids can be used by other tissues besides the brain and the red blood cells. The low insulin-glucagon ratio during this time of early starvation also causes another very important effect, and that is the transport protein that we find in adipocytes as well as muscles. This transport protein is called GLUT4 for glucose. This transport protein during this low ratio will actually be found inside the cytoplasm. It's not on the plasma membrane. So if it's not on the plasma membrane, these cells, these muscle cells and adipocytes cannot take up glucose. And if they cannot take up glucose, that means this glucose will be reserved more for the brain as well as for the red blood cells. Note that gluconeogenesis, the synthesis of new glucose that we discussed in the last video, also begins during early starvation and helps maintain the blood glucose concentration. Now, gluconeogenesis in the liver requires ATP. So you can see to make one glucose, we need 6 equivalents of ATP that we discussed in the last video. So the question is, where does this energy, this ATP, come from during this state of starvation? And the answer is fatty acids. So the liver, just like many other cells, including your muscle cells, as well as your adipocytes, they all use fatty acids to generate energy. So the liver will carry out beta-oxidation and generate ATP. It's more than sufficient ATP, and this ATP can be used to make glucose by gluconeogenesis. As we discussed in the previous video, beta-oxidation of fatty acids releases not only acetyl-CoA but also FADH and NADH, and all of these things, the acetyl-CoA goes through a Krebs cycle, the FADH2 and NADH enter the electron transport chain, and all of them can eventually give you considerable amounts of energy for gluconeogenesis in the liver. Another thing that gluconeogenesis requires is building blocks, raw materials. So we need building blocks to make glucose. As we discussed in the last video, this building block could come from many different sources. For example, lactate, pyruvate, oxaloacetate, glycerol, or anything that can be made into intermediates which can be converted into glucose. Lactate is an important building block during this time for making glucose, and this lactate can come from red blood cells, since red blood cells carry out glycolysis to generate ATP. They don't have mitochondria, so they are continuously producing lactic acid, and that lactic acid can go to the liver and be used by the liver to make glucose via gluconeogenesis. Then there are glycolytic cells, such as the renal medulla. Although they have mitochondria, these cells do not have enough oxygen due to the blood supply anatomy. So since they don't have enough oxygen, they also generate quite a bit of lactic acid, and that lactic acid from these glycolytic tissues can be used by the liver to make glucose via gluconeogenesis. Research also shows that considerable amounts of lactic acid can come from muscles as well as the GI tract, and this lactic acid once again can be used by the liver for gluconeogenesis. Another building block for gluconeogenesis is glycerol. Glycerol can be converted to glycerol 3-phosphate and then into dihydroxyestone phosphate, and this is an intermediate for gluconeogenesis. Glycerol comes mostly from lipolysis, breakdown of triglycerides into fatty acids and glycerol that happens in adipocytes. So during this time of early starvation, the building blocks for gluconeogenesis, liver gluconeogenesis, will be lactate, glycerol, as well as pyruvate, while the ATP, the energy required for gluconeogenesis, will be fatty acid beta-oxidation in the liver. So just to summarize, in early starvation, most of your glucose will come from glycogenolysis in the liver. This is the glucose that you find in your bloodstream, while about 25% of the glucose will come from gluconeogenesis. Now let's see what happens in late starvation. So some of the major events that we will see here will be proteolysis, that is muscle protein breakdown, excretion of urea, gluconeogenesis, and lipolysis, as well as beta-oxidation of fatty acids. As we mentioned earlier, after 1 day, all the liver glycogen will be gone, and we enter this state that we call late starvation. So since there is no more glycogen present in the liver, now glycogenolysis cannot maintain our blood glucose or contribute to maintenance of blood glucose. So now gluconeogenesis in the liver will have to maintain all of this glucose that we find in our bloodstream. So as we mentioned earlier, gluconeogenesis requires two very important things. One is energy, ATP, and that's not a problem because the liver gets energy from fatty acids, and we have enough fatty acids, triglyceride storage that will last us for weeks, if not months. The problem is, so therefore, not the energy, the problem is the building blocks. Okay, so where do these additional building blocks come from? In order for gluconeogenesis to increase in the liver, so that now it is the only process, not glycogenolysis anymore, only gluconeogenesis is maintaining our blood glucose at greater than 40-50 milligrams per deciliter in order to feed the red blood cells and the brain. So besides pyruvate, lactate, and glycerol, the additional building block will now come from muscles. So we'll see that muscles will provide the additional building blocks to the liver for gluconeogenesis. The decreased insulin-glucagon ratio will cause muscle proteins to break down. And when these muscle proteins break down, we form amino acids. And these amino acids are converted into mostly alanine and glutamine. And the liver will use especially alanine, predominantly alanine, although it may use other amino acids, but it seems that it prefers alanine. And it will take this alanine and use the carbon skeleton of alanine as the building block for glucose. Now, since amino acids have amine groups, the amine groups are removed, the amino groups are removed, and in the liver they are converted into urea, and that nitrogen is then excreted by the kidneys as urea. A common finding in research on starvation shows that the branched-chain amino acids, valine, leucine, isoleucine, BCAA, the concentration in the plasma tends to increase during the starvation period. So research shows that BCAA, the branched-chain amino acids, may be an important source of alanine and glutamine and for gluconeogenesis. So once again, the alanine can be used by the liver to make glucose. So besides gluconeogenesis in the liver, more recent research has shown that even the intestine, the small intestine, the enterocytes of the small intestine, they can also carry out gluconeogenesis. And they seem to prefer glutamine rather than alanine. So the glutamine is transported to the small intestine, and there the carbon skeleton once again will be used to make glucose, while the amines will come out, in this case, as ammonia in the intestine, and this ammonia will be sent to the liver. There it will be converted into urea, and the urea will be excreted by the kidneys as urea in the urine. So during this time, the gluconeogenesis that is happening in the liver as well as the intestine will be maintaining our blood glucose, and the excess nitrogen that is coming from the breakdown of amino acids, that nitrogen will be converted into urea and excreted into the urine by the kidneys. So as starvation continues, we get into a later stage that's called protein conservation stage of late starvation. And during this time, we see the major metabolic events that happen include ketogenesis, that is the formation of ketone bodies, proteolysis, muscle proteins are breaking down, but at a reduced rate. There's less muscle protein being broken down than the previous stage. A lot of the nitrogen is being excreted as ammonium. Gluconeogenesis continues, as well as breakdown of triglycerides into fatty acids and beta-oxidation. So we cannot keep on breaking down muscle proteins because the starvation, the late starvation state, means we are probably lost in the woods. We need to find food, so we have to move around, and if we keep on breaking our muscle proteins, we cannot move around to find our food. So in this case, we now see the formation of a new fuel molecule, and this is ketone bodies. So why does the liver form ketone bodies? Well, during the starvation state, remember the liver is getting energy from fatty acids, so there's a lot of acetyl-CoA that is formed. Now, this acetyl-CoA, these large amounts of acetyl-CoA, unfortunately cannot enter the Krebs cycle because oxaloacetate in the Krebs cycle, this oxaloacetate is being used to make glucose by gluconeogenesis. Okay, so since more and more oxaloacetate is being diverted towards formation of glucose, this acetyl-CoA cannot enter the Krebs cycle, and so what happens, the acetyl-CoA will go into this formation of ketone bodies. They will combine with one another to form ketone bodies. So how do we form ketone bodies? So one acetyl-CoA will combine with another acetyl-CoA and to give you acetoacetyl-CoA. This reaction is catalyzed by the enzyme thiolase. And in the next reaction, catalyzed by HMG-CoA synthase, the acetoacetyl-CoA will combine with another acetyl-CoA. And now that gives you this HMG-CoA. And the enzyme HMG-CoA lyase will remove one acetyl-CoA to give us acetoacetate. So acetoacetate is the first ketone body. But remember, during this time, a lot of fatty acids are being broken down, so there's going to be a lot of NADH around, a very reducing environment. So in the presence of a lot of NADH, this acetoacetate, this carbonyl group here, can get reduced to an alcohol, giving us the second ketone body, beta-hydroxybutyrate. Now, some acetoacetate can also undergo decarboxylation, carbon dioxide. This carboxyl group can be lost as carbon dioxide to give us this volatile ketone body that is called acetone. So these three ketone bodies are formed by the liver, and they will be transported out of the liver into the bloodstream by the monocarboxylic acid transporter, or the MCT. A few important points to note about ketone bodies. One is that the predominant ketone body typically found in patients is beta-hydroxybutyrate because of this reducing environment, the large amounts of NADH. It's important to note that not all these three ketone bodies are ketones. This is a ketone, this is a ketone, but beta-hydroxybutyrate is not a ketone. So when we test for ketone bodies, we have to be careful because there are some tests that only test the ketone part of ketone bodies. Those tests will not pick up beta-hydroxybutyrate, which happens to be actually a major ketone body that is typically found in patients who are on starvation or even diseases like diabetes. Fortunately, now we do have a specific test for beta-hydroxybutyrate, and so it's important that we use this test in order to detect beta-hydroxybutyrate, which might be the only ketone body present in sufficient amounts or large amounts in a patient. Another problem with this term ketone body is that usually this term bodies imply something very large. Okay, however, ketone bodies, remember, these three ketone bodies are very small structures, and they are very water soluble, so they are readily filtered by the kidneys. So they're really not bodies, but they are ketones. Only two of them are ketones, this and this, while the third one is an alcohol. Small water-soluble chemicals. So that's another thing we see during the starvation process is the concentration of ketone bodies increase in the bloodstream. As we mentioned earlier, the major ketone body is beta-hydroxybutyrate. There is less of acetoacetate that is usually found. Acetone is usually not found or found in very, very low concentrations. Since acetone is volatile, it can escape out through the lungs. So what's the advantage of producing ketone bodies by the liver? The ketone bodies that is produced by the liver get out into the bloodstream, and now these ketone bodies can be used by the brain for energy, for ATP. So now the brain not only has glucose for ATP, but it also has ketone bodies. Of course, these ketone bodies can also be used by all other cell types, but not by the liver. Okay, the liver cannot use ketone bodies, neither the red blood cells. So except for the red blood cells and the liver, everyone else can use ketone bodies to generate energy. How are ketone bodies broken down to provide energy? Well, remember, ketone bodies came from acetyl-CoA. So basically what we get at the end is acetyl-CoA when we break down ketone bodies. The beta-hydroxybutyrate is converted into acetoacetate by this enzyme here, and then acetoacetate is converted to acetoacetyl-CoA by this enzyme SCOT. This is the enzyme that's not present in the liver, so that's why liver cannot use ketone bodies. Eventually the acetoacetyl-CoA is converted to acetyl-CoA. So basically, acetyl-CoA can enter the Krebs cycle and provide the brain, the heart, and most other tissues except your liver and your red blood cells with ATP. So ketone bodies now become a very important fuel for energy generation. So with ketone bodies present now, the brain uses much less glucose, from 120 grams a day at the beginning to less than 40 grams a day of glucose, because now the brain can use ketone bodies. As starvation progresses, besides the liver, now the kidneys also start carrying out gluconeogenesis. Like the enterocytes of the small intestine, the kidneys also prefer glutamine as the source of carbon skeleton. So glutamine carbon skeleton is made into glucose via gluconeogenesis, while the amine groups of glutamine will come out. The amino group will be released as ammonia, and that ammonia will be excreted by the kidney as ammonium salts. So as kidneys start their gluconeogenesis, the liver gluconeogenesis goes down. Okay, so liver gluconeogenesis decreases as kidney gluconeogenesis increases. Now more and more urine nitrogen is in the form of ammonium rather than urea. Now ketone bodies are very small and water soluble, so when their concentration is high, some of these ketone bodies will be lost in the urine, and we can detect ketone bodies in the urine. They will be combined with ammonium salts. The ammonium is also excreted by the urine, so we'll usually see ammonium salts of hydroxybutyrate or ammonium salts of acetoacetate. But since ketone bodies are very important fuel at this stage, the kidney will slowly start reabsorbing more and more of these ketone bodies, so that the amount that is lost in the urine decreases. So as starvation continues, we eventually reach a steady state. The blood glucose concentration is either normal or on the lower end of normal. The beta-hydroxybutyrate, the ketone body concentration, is pretty high, around 10 millimolar. Bicarbonate concentration is 18 millimolar. This is needed to neutralize the acidity of the ketone bodies, which are basically acids, hydroxybutyric acid and acetoacetic acid. The normal bicarbonate is 24, so some of this bicarbonate has been used up to neutralize these ketone bodies. So at this late stage of starvation, the brain will be getting almost half its energy from ketone bodies and the remaining half from glucose. The kidneys will be using fatty acids and ketone bodies. Red blood cells will continue to use only glucose. They cannot use ketone bodies or fatty acids. The other cells, such as your muscle cells, as well as the adipocytes, will continue using fatty acids and ketone bodies. Remember, ketone bodies, beta-hydroxybutyric acid and acetoacetic acid, these are weak acids, but when they are present in very large amounts, they can lower the blood pH and cause what is called ketoacidosis. And this drop in blood pH can be a very dangerous situation and can lead to fatality. So this is the answer to the question, what happens as we go from having our dinner and then not eating for several days. It is indeed remarkable how our organs work together to keep us alive during the prolonged starvation states. We'll continue in the next video a little bit more on late starvation. So until the next video, thank you once again for watching. Have a wonderful day. Have a happy and healthy day, and we'll see you soon.